Hi, everyone. Can I have your attention? We're about to begin, but before we do any of the curators have any announcements? Go Tigers! Uh, all right, then it's my pleasure to introduce Dr. Henry Bart. They are a professor of ecology and evolutionary biology at Tulane University, where their lab focuses on the taxonomic and ecological diversity of freshwater fishes. They also serve as the director of Tulane University's Biodiversity Research Institute and the curator of their fish collection. But before joining Tulane, they got their Bachelor's of Science and Master's of Science in Biology at University of New Orleans and a PhD from the University of Oklahoma in zoology. And they also served as faculty at the University of Illinois in Auburn. So here to tell you a little bit more about the work they've done in their impressive career is Dr. Henry Bart. So give him a round of applause.
Thank you all for inviting me. I just want to make one point of clarification. I'm retired. No, I used to curate the fish collection and used to be the director of the museum, but thankfully there's someone else that's doing that now. So I can enjoy my retired life, really retired life. And I'll have more to say about that too. Let's see. There we go. Oh. Yeah. Okay. So the first thing I want to do is just tell you a little bit about myself and the things in my career I don't get to talk about enough until we'll get to the topic of a little bit later on. But I was hired as an assistant professor of EEB and curated fish collection back in 1992. And a year later, I was appointed director of what was called the Tulane University Museum of Natural History. I replaced the gentleman pictured here on the right, Royal D. Sutkus. He's the one who built up the fish collection to its ginormous size. When he first came to Tulane, there was only 14 lots of fishes left over from a very early university museum that I'm going to say some things about later as well. And in the time during his career, he built it up to a collection over 208,000 lots of fishes and over 6 million specimens. And that's what they had when I took over as curator. During a career spanning 55 years at Tulane, which tragically ended because of Hurricane Katrina, his field sampling, I mean, he survived. But he made more than 10,000 field collections and had a hand in collecting over 5 million of the specimens in the collection. So that just shows you like one person's contributions to collecting. My contributions, on the other hand, in field collections and specimens was much more modest. I haven't even reached 2,000 field collections. I can't really tell you how many specimens I collected, but I was determined not to repeat a collecting effort like Sutkus did. Because for one, we didn't have the space to accommodate it in the buildings we occupy. And, the other issue was I didn't really feel like killing all of those fish, okay? So my goal, my mission was to make sure we use those specimens. And this is part of what the talk is about, is the technological ways that we are trying to do that, to make sure those specimens didn't die in vain. I'm obviously going the wrong way. All right, so you really need to see the Sutkus fish collection to appreciate it. It is literally jaw-dropping. I don't know how many. That allows you to look down these 23 double rows of 40 feet shelving, 13 feet high. I look at the people that I'm showing it to, and their jaw is literally hanging open. It occupies 30,000 square feet of floor. The one shown on the right are these World War II vintage ammunition storage buildings that functioned during World War II. The buildings were designed to hold naval ammunitions. They're virtually indestructible, and that's why we existed there for so long. We now have over 8 million specimens because we acquired 78,000 lots and over 1 million specimens from the University of Louisiana Monroe. That collection had been orphaned, and the university was threatening to throw it away. So I organized a group of regional curators, and with NSF Rapid support, we brought the collection to Tulane. I had hoped to give much more of the specimens that we took into other museums. That hasn't happened yet, but we will continue. Actually, my successor will continue, trying to get people to take some of those collections, or we will send them off for exchanges and things like that, which is a very, very good use of that material. Okay, so at one time, Tulane had this university museum, and it occupied the upper floor of the main administration building on campus called Gibson Hall. And it was amazing. It had these galleries of exhibits that came to Tulane from the World Cotton Centennial Exposition that was held in New Orleans from 1884 to 1889. Tulane purchased those collections and put them up in what became its administration building. You can't really see this now, but Nick, this is the diorama I mentioned. That's a tree trunk, and it's got 7 ivory-billed woodpeckers on it. It's an amazing sight. It's a black and white photo, so you can't really see the birds that well. But I mean, we had treasures like this in the University Museum for a long time. This was way before my time at Tulane. The museum survived. I'm sorry, I'm having a hard time with the switch. We had collections of birds that my predecessor added. We had trophies of different kinds of animals. This upper left side image is an image of a family of platypuses that were on exhibit in the Cotton Centennial Exposition, and that came over to us, but the exhibit itself was falling apart. The diorama was falling apart. Amphibians and reptiles, and of course the fishes. Thankfully, all of these other collections went to LSU, and in fact are part of the museum here at LSU. And it's a good thing because the collections, for pretty much the time I was hired until we decided to donate them to other universities, they were not being curated. And if they weren't being curated, they couldn't be used like they needed to be. And there wasn't no one to really raise money, for taking care of them. And so we were very happy that LSU had curators for all these different kind of collections and willing to accept them. And I'm sure that the specimens are getting a lot more use than they would have had they stayed at Tulane. Okay, so I started Tulane in 1992. That's me in 1992. This is me today, a little thicker, a little older, a lot wiser. Thankfully, Brian Siglauskas, who you guys met in a seminar he gave last year, was hired to replace me as both director of the Institute, TUBRI, and also curator of fish collection, and he's also a professor of ecology and evolutionary biology at Tulane. And so all is good. I mean, this is what we want as collection curators. We want the collections that we took care for how many years to live on and to be used, by the research community. And in too many instances, like the University of Louisiana Monroe, collections get orphaned and abandoned, and someone has to rescue them. So that situation is not affecting us. Okay, so in 2011, we changed Museum of Natural History to the Tulane University Biodiversity Research Institute. This was after all of our collections were donated to LSU. We were given additional resources for the fish collection, for taking care of the fish collection, including another curatorial assistant. Our mission changed to biodiversity informatics tool development. I want to talk to you about that briefly. Technology for improving and sharing biodiversity collection data and with other institutions. Technology for using our specimens in research. And that's like the main thing I'm going to talk to you about today. So here's some examples of the early technology projects. I'm sure you've heard of these. So geolocate was created at Tulane, actually before we we changed our name to Tubri. And that's because of the gentleman shown in the little cutout picture, Nelson Rios. Nelson left us 18 or 18. He's now at Yale University. And, he's continuing the work he did with geolocate, but for 17 years, it existed and was funded at Tulane. We also, in 2009, we took over FishNet2, which is a global network of fish collection databases. 81 fish collection databases currently, provides research access to about 5 million specimen voucher records. By that, I mean lots. It represents more than 50 million specimens from all over the world, and that the lighted parts are all the different data points in FishNet2. And then we projections to 2099, okay? And so you could go to this platform and you can get data on those kinds of stream flow and temperature conditions from any place you specify in this data option over on the right-hand side, okay? What we want to do with this, well, what someone is going to do with it, I hope, is add additional environmental layers so that, this thing we'd be able to about lots of things about fish, more things about the fish that occur in some of these same places that are collected in in a lot of the same rivers. But we could do this for other kinds of environmental variables. So we could even predict, what kinds of morphology or something like that specimens have based on this kind of data. And so this is hosted, all of these projects, well, except geolocate. Now that's moved to Yale, but the others are actually hosted on servers, which are maintained as part of Tulane's IT infrastructure. All right, so the taxonomic impediment, which is what I really here to talk about. So this is a worldwide shortage of taxonomic expertise, especially taxonomists that are capable of dealing with the most poorly known groups of organisms. The result of this is that there's gaps in knowledge of species diversity. This is potentially disastrous consequences, efforts to conserve species and ecosystem services. And this is what's called the biodiversity crisis. Okay, so these are all the species that I played a role in describing, about 24 species. Actually, they're not all fish. There's a couple of aquatic salamanders, because I study them too. But there's actually twice this number of species that I'm still sitting on, okay? And the reason why I haven't described them is that it takes a long time to describe a species. It takes a long time to visit all the museums where different, specimens of those species are and make counts and measurements on them, and then bring the data back and analyze it, only to find that, the traits aren't necessarily that good for taxonomic diagnosis. So, we need to come up with another solution to that problem. Oops. Okay, that's what I said. All right. There was a study published in 2011 by Mora et al., which estimated that planet Earth is inhabited by 8.7 million eukaryotic species, give or take 1.3 million. But that's a lot more than the species that we have described to this point. Taxonomists working since the time of Linnaeus have only described about 2 million species, only 23% of the Mora et al. Predicted number. An estimated 900 species go extinct each year. Climate change is accelerating the rate of species loss. Thus, we're at risk of losing most of the world's biodiversity before it can even be discovered and described. That is the biodiversity crisis. All right, so there's a study that I was talking about. How many species are there on Earth and in the ocean? These authors showed that higher taxonomic classification of species, that is to say, the assignment of species to higher taxonomic categories like phylum, class, order, family, and genus, follows a consistent and predictable pattern from which the total number of species in a taxon is estimated. And that's what they did with these plots here. They used these plots showing accumulation curves of the number of species as you go across all these different hierarchy. And from that, they generate these predictions about the number of species they are. And so there's the group that I studied, the Actinopterygii, and their estimate is that group has about 100,000 species, and we've only described about 32,000 to date, based on the number of valid names. All right, so this is the point about fish species diversity, only 32,000 described species. Moredell predicted the number of ray fin fishes alone would be 100,000 species. At the rate taxonomists are presently describing new fish species, it would take 340 years to describe all the fishes predicted to live in the world's rivers, lakes, and oceans. The problem is that traditional methods of fish taxonomy are laborious and often don't yield data that's useful for species diagnosis. So how do we fix that? Okay, well, thankfully for me, at a pretty late stage in my career, I became involved in two NSF-funded projects. One is Biology Guided Neural Networks. That was the first one. The second one was called Imageomics Institute. Those grants involve all the people you see here. You probably can't read their names or any, because the text is kind of small. There's obviously a lot of people and a lot of different organizations and institutions involved in the project. The premise of this project is that we can train computer algorithms to extract biologically meaningful data from images of organisms. My group and collaborators that we work with at Drexel University and Virginia Tech are using machine learning methods to process and segment features in images of fish specimens, extract trait data from the images, and explore expedited methods of taxonomic discovery. We have developed a new method of trait data extraction that we call morphological barcoding. It's like DNA barcoding, but it involves morphological data. It's a relatively new topic. We really haven't published anything on it yet, but I've been talking about this at several conferences I attend. Okay, this is a very busy slide. I'm going to walk. You can't undertake a project like that without building infrastructure to host all the images, to make those images available, you have to have a workflow to do all of the machine learning tasks and to make sure those tasks are performed correctly. You have to gather the images of organisms, and that's what the slide is showing. We've processed today about 114,000 fish images. There are 6 data sets from 18 source institutions, mostly museums. In other words, other museums did the imaging for specimens that we're using in the project. We have 13,558 species of fishes, of teleos. That's 42% of described teleos. The images represent 3,500 genera, 500 families, very close to the number of fish families. They're color images, if you can call them that, because most of them are museum specimens. They have a very clean background. We try to limit ourselves and images that have clean background. They're standard left side lateral views of fish, like shown on the right page. And they're whole fish. If we had an image where part of the fish was cut off, we didn't use it. Or we flagged it as low quality because not having a whole fish affects how the algorithm does the kind of performs the task that we want them to. The average image size is about 6.6 megabytes, which is a pretty good size image. And we have image quality metadata for about 38,000 images. And the beauty of that is that you can look at those image quality metrics and decide whether you want to use that image in a project or not. In fact, we have measures that show how likely the results would be affected if you use a low quality image. So the workflow just works like this. On the right-hand side, we first use object detection. This work is done at Tulane and Drexel Metadata Research Center. The object detection finds the fish in the image and removes it from the background. We also take things like scale bars, which we need, and any labels that have metadata associated with those images. But because it's in museum specimens, the databases at the different institutions also have very rich metadata about the specimens that we use. And collaborators at Virginia Tech, the Carpatnia Lab, segment certain of the body features in these different colors shown in the image on the lower side. And these are the data that we use, some of the data that we use to do these different morphological comparisons. I mentioned that the other organizations in the imaging, the Great Lakes Invasives Network was involved. There's like 5 different ichthyological collections that participated in that project. We also got images from GBIF and iDig Bio, the digitization hub of NSF's Advancing Digitization of Biodiversity Collections program. This infrastructure that we built just on how we process and manage images is in a web accessible project called Fish Air. And there's the URL and there's the images. Remember, this is a work in progress. We have a pending grant that, if funded, will allow us to completely build out this thing. And the way this is designed, I think I can say this now, I'm gonna get ahead of myself a little bit. The way it's designed is that you can image your own specimens and you can put them in this platform. And then we can perform the machine learning task for you if you're a researcher and want to use this platform. And so you get to study the fish that you're interested in studying that way. All right, so the hypothesis of morphological barcoding is that neural networks can be trained to extract new kinds of more diagnostic trait data from 2D images of organisms. I mean, it's almost unbelievable because this is just 2 of the 3 dimensions, right, of the specimen. And yet, I'm going to show you, we can get really good data from those images. Not just fishes. This can be applied to anything. I think it can be applied to plants too, but I just haven't tried it. But the idea is that we can do this very rapidly, and the trace will help taxonomic discovery. That's the theory. We do things like feature segmentation. I showed you where we highlighted the fins and the eye and the head and whatnot on that fish. We could also compute the centroids and then the areas of those features. This is all part of methods that we developed using landmark data, simply landmark data. We even figured out how to automate the process of landmarking the image. I'll say more about that later. We use color palette data. You're going to be amazed. We can generate these histograms from RGB and grayscale colors that we just take from the image. We can take little strips of pixels. We can say, give me a strip of pixels, 1 pixel wide, X number of pixels long, from some character-rich feature of the fish, and we can get data. We get that data from an image. We also use these outline methods formed by what's called Freeman chain code methods and closed curves and Fourier transform methods. And I'm going to show you examples of all that. All right, so here's an image of a segmented fish. This is Notrompus eriomus. These are landmarks that were placed on that specimen by an algorithm, a computer algorithm. We estimate that, well, let me show you some animations that are here. I'm sorry, getting ahead of myself again. We can look at just the landmarks alone could give us a sense of geometric shape variation, okay? But we can also compute interlandmark distances, and we can compute areas where 2 landmarks overlap, they intersect, I'm sorry, and how, am I going the wrong way? And how, oh, hitting the wrong button? There's something else supposed to animate. It's all right, I'll describe it to you. I can describe it. Where these 2 interlandmark distances intersect, they're angles that are formed, okay? The question is like, what does that tell you? Well, we don't know. It might tell us something really interesting. It's data that we can derive from those images. Just the landmarks alone, we estimate can generate data for 20,000 images. 20,000 characters, I'm sorry, characters. All right, that's the other thing I was trying to show. Thank you, Amber. Help me with that. All right, color palettes. Here it is. Now remember, these are drab museum specimens, right? And so you say, I want you to give me some color data, a color palette for that image. All the different colors per that image. You can ask a computer to do it. It will generate something like this, okay? Histograms. Now, all of these, what this is saying is that all of these colors shown on the right-hand palette are from that fish. And if you look down to the fish of sunfish, there's obviously different groups of fishes, right? But you can see the palettes, how they differ. The two sunfish are lighter, they have lighter colors. The minnows are darker, they have lighter colors. But look at the histograms. Here's the RGB color histogram for the first sunfish, and there's the grayscale histogram. Here's the one for the other one. Minnows. Again, the histograms, both RGB and grayscale, show the same patterns because there are similarities in those two groups of fishes that allow you to group them that way. Here's an example of taking strips of pixels from images. So let's say we take these two fish. One's on the right is a maronid, an anadromous fish. Obviously a different group of fishes because it's got spiny rayed fins and soft rayed fins. So if we say, well, okay, give us a strip of pixels from like there, top to bottom on that minnow, it gives us this little data right here in that segment to the right. And then take a strip right across the lateral line, and it gives you that. And then take a strip of pixels, same thing, horizontally on this maronid, it gives you that. Take another strip, it gives you this, okay? So with that data, we can distinguish those different groups of fishes. I might say those different families, that's not a real challenging problem. It isn't, but we can take pixels like that from anywhere on those images. We can derive that data from those images. And my belief is that this is biologically meaningful data because the data is what's coming from those specimens. And these just show that, the strip at the upper part of this minnow is catching that silvery bar that it had on its side. And this one on the maronid, I'm not doing this right, but this little area right here, you can see it, is catching, it's detecting that trait, that it has stripes, whereas the other one has a silvery band. Okay, there we go. All right, here's another case. We can train algorithms to derive scale counts from pixel strips taken from the lateral line. Two different Gambusia specimens. The white box is where we said to take the pixel data. The two other diagrams below it are color and grayscale pixel strips from that area. And the reason why it has this pattern is because of the pigment outlining the scales, okay? So we can generate from this bar chart or these peaks. And if you count the scales here, you count the peaks, blue and orange, they agree by about a scale. So we can teach the algorithm how to count scales. It won't take us hours counting scales. And as long as we have big enough populations of these specimens, do in my collection, we can generate, that kind of data, and we don't have to spend so much time getting it. Okay, the outline methods, as I told you, just involve like outlines, data you can get from outlining the this like a silhouette of the fish specimen. Different methods are called changing slopes of points and closed curves. And by the way, a lot of literature on this is done with plants and the leaves of plants, how do people study plant leaves. These things extract slopes from a curved line, which in our case are slopes from the outline of the fish. So we can get data from the outline of the fish. The fish has one fin in the middle of the back. We get data when that curve goes up to go around that fin and actually traces the contour of that fin. We can get, if the fish has two dorsal fins, it gets a signal produced, by both of those fins as it traces that contour. The other method is called Freeman chain codes. It outlines are formed by selecting a starting point on an object's boundary and recording a sequence of directional codes to trace the perimeter of the fish. The process concludes when the tracing returns to the original starting point, creating a closed loop. And that data is also data that we can use in this process. The third method is called Fourier transform in PCA. The one that we use is called elliptical Fourier analysis. This describes the entire closed contour of a shape with a series of harmonic ellipses, providing a comprehensive landmark independent method for shape quantification. And this is particularly good for complex irregularly shaped objects or when corresponding anatomical features can't be identified. All right, so some examples of this. Here's those similar comparison of the two minnows and the two maranids. And what you're looking at here is how the curves, which are traced in a thin black line around each of these fish in the image, how they generate data and how that data helps to distinguish them based on features of the snout. And you can see differences in the snout in these two groups of fishes, based on pectoral fin differences, anal fins, and dorsal fins, okay? And so these are signals, this is signal data that we can get using that kind of changing points on closed curves method. And then when you use outlines formed by closed curves, similar thing. The outlines look like the fish images. That's because they're from the fish. And again, we can distinguish here, in this case, two groups of minnows, two different species of sunfishes. And if we add PCA to the analysis, we can separate those. We can separate the minnows, two minnows, from the sunfish. And again, I'm not saying that's a very challenging problem. It isn't. But if it's an easy thing, but we can get down into much more difficult tasks by comparing the species in pairwise ways. Okay, so in order to do this, to generate barcodes, we need a minimum of 50 images per species to train the ML machine learning models. 30 is in the training set, 30 images in the training set, 60 are to test the performance of the model. And you really want a high rate of success with performance before you accept the result, like 95% or better. Barcodes are going to be based on the smallest subset of characters useful to distinguishing paired species. We have to compare every pair of of fish species in order to accomplish what we're trying to accomplish here, which is a lot of pairs. And, we have to make sure we have enough images for that. And, we're sure we don't in this data set because, even though we have large numbers of images, we don't have every species in our data set represented by number of species, the right number of images. This is why having other people image and contribute data sets is going to increase the performance of the platform. We are confident that our barcodes can distinguish families and genera, as I already told you. Species is going to be much more challenging. The proof of concept is going to come from a data set of North American minnows from the family Mucidae. Anyone ever try to identify a minnow knows how challenging it can be. We're getting a pending STAR proposal that we want to prove that we can do it. But like I said, additional image is going to be needed to cover all fish species. Okay, here's an example of a morphological barcoding base. These are 10 species of minnows. You can't really read the names. But one of them is the minnow I showed you, the landmarks, Metropus areolus. Turns out that thing is like different from all the minnows we compared. And the other one is a mineral called Neutropus percobromus. And we discovered on the landmark data. This is only landmark data. So the scatter plot of characters, the scatter plot shows characters CH18, CH10 here on the x-axis and CH18 on the y-axis, okay? This is from shape data with 10 different species of Neutropus, but these aren't really distinguished that well. Here's how we generate the morphological barcodes. So the morphological barcode for Anariomus can be defined as, morphological barcode Anariomus, character 18, 67.5 on the axis, 1,150. The morphological barcode for Percobromus, which distinguishes Percobromus from the other minerals, is character 10, 0 to minus 15, point 15. Character 18 data to distinguish it is 0, 67. Okay. So again, that's not a challenging task. It's a pretty simple task because we can easily separate the species based on their shape. The more challenging thing is to separate this kind of cluster and then to separate the other 280 species of minerals in North America. But I'm confident that we can do it. Okay, so here's the part in the talk where I have to acknowledge a team of very smart computer scientists that have been working with me. The one that deserved most credit, the whole concept, is fellow just below me, Yasin Bakish. He is Senior Manager of Biodiversity Informatics and Data Engineering at TUBRI. Below him is Xiaojun Wei. She is a programmer, and she separates all of our infrastructure like databases, servers, web servers, and things like maintains them, and service them when needed. And then Mahir Tintas was with us. He just went back to Turkey. He's Turkish, like Yasin. He just went back to Turkey. And we told him if we needed him, we would hire him as a contractor. He came up with that really nice object detection protocol and trained the algorithms and everything. He's actually, by training, he's a mathematician and a chemist. And he's been doing amazing work. I also have to acknowledge my collaborators at the Metadata Research Center, Jane Greenberg, and her students at Drexel University. Anuj Kompatny in his lab, he has what's called the Knowledge Guiding Machine Learning Lab. He's at the Tech. All of my Imageomics Institute collaborators for helping me understand and appreciate computer methods of image analysis. It's not my area of expertise. I'll admit the first one to admit that. But these are powerful methods, and I have a lot of faith in them. And it's really great interacting with those people. We just had what's called an HDR ecosystem meeting in Columbus. And it was great interacting with people from all those different HDR Institute projects. We had help with graphics for things like the fish air illustration. And I also want to thank all of you for your attention. Thank you. Happy to answer any questions. Yeah.
Thanks a lot.
You're welcome.
Yeah. You described as a really important goal in all of this, the acceleration of taxonomic discovery. And so what I'm wondering, I get this.
You get what I was saying?
I get what you're saying. For the things that we've already named, we already know. So is it possible that there would be a connection to the giant, photo database that's being generated by citizen scientists through iNatural sorts of things? Yeah. Pour it into to do the discovery part.
This could all become part of it.
Okay.
And this is why. So in imageomics, one of the really big discoveries was something called foundation model that involved, I think it's 10 million images from iNaturalist. the concept of a foundation model is you give a computer algorithm images of different kinds of things. You don't tell it anything about the images, you don't give it any metadata. It processes those images, it identifies the things that are common to the image, like a bird in the image, right? The 2 or 100,000 images. And then it learns, you can train it to learn, just with the images, how the different birds, doesn't know they're birds, it just knows they're the same in terms of how they look, how they differ from one, okay? It doesn't tell you that though. It just does a good job classifying species. And so it's called BioClip, by the way. You can all play with it. You can give it your own image and see if it identifies correctly. Any animal. I think it's trained for animals, not plants. That's why I have it with my house by plants. But I'm not saying this is not good. But your point is well taken. The problem with foundation models, they don't really tell you, except that, oh yeah, I figured out this task and I did. And you can give me any picture you want from a universe like iNaturalist and I'll take it. Okay. What we need is something called a different kind of AI called explainable AI, interpretable AI. And what that does is it tells you how it did it. It tells you what it used. So that's where that kind of data comes with explainable, a different kind of a model. It's not the same as the foundation. It's an add-on to that. I think it's kind of controversial, but who cares? We're going to learn, right? If it's explaining something and you can prove its biological meaning, that's weird, right? Then who cares?
That's great. So with your minnows, you're focusing a lot on the lateral left side. And I'm wondering if you've thought it on in other fishes, like a benthic fish, maybe, where it's like dorsal naturally.
It's Latin.
Yeah.
Flounders.
Yeah. How would that work?
So flounders are going to have to be oriented differently, and models have to be retrained, right? Because they're really kind of on their side, but their dorsum is like the top and ventral is the bottom. I don't even know how to describe it. But yeah, so that's and then you have to do a lot more of it. Because the fish, there's platycephalus, which also, if you look side to side, it looks like this. It doesn't look like a fish at all. We have to train the model, right? And then it will segment the features. I don't think it's gonna be a problem. Well, we have to train all the models in our work to recognize that, oh yeah, this is a fish, but these are the different features. So what we here's another thing. A flounder has like one big continuous dorsal fin. And the anal fin is pretty continuous too. It kind of like wraps around both sides of the body. And so we're gonna have to have it, but you can actually, they don't have, some of them have only one fin. Some of them don't have the other. See what I'm saying? There are a lot of exceptions, and there are probably a lot of exceptions to rule. We're starting with minnows because we want to show that we want to convince a bunch of skeptical freshwater ichthyologists. Then we're going to tackle bigger problems. So listen, I'm not going to be the one driving this. Okay. But I'm fine with it. I just want to see it happen. I want to discover it. And so whatever we need to do, I mean, I can see lots of projects, lots of money coming to this if we just prove it. When we convince taxonomy, skeptical taxonomy. That's the question. Yeah.
So really cool talk. For some of these fish, so for most of this seems like it'll be really outstanding. Obviously there are problems. Are there plans to try and use these models to extract sort of incomplete data to be able to say, hey, there are a lot of things that are messed up here, but here's my best guess. So for instance, what I'm really thinking of here is deep sea fishes, because you know as well as I do that when they come up, it's a great day if they're in good shape, and they can be ridiculously sexually dimorphic, and in many cases we have no idea what the hell they're supposed to look like anyways. So we could be looking at troll trauma, we could be looking at dimorphism, we could be looking at almost anything. And yeah, as you said, these are like the model is still being trained. And if a specimen is smashed, then okay, you're probably not going to get much out of it. But I'm wondering if there are any thoughts at this point about extracting incomplete data sets to get people looking in the right direction. Because for those deep sea fishes, I feel like that if you can get it to the point where a machine can guess and then you can get some human intervention, then you might be able to make a lot of progress with a specimen that otherwise would basically just be junk.
So deep sea fish are challenged like that.
Yeah.
Because they can be bizarre, especially look at that block there, like, you know what? You know, it's all a function of model training. Like, sexual dimorphism. You know, their difference. The foundation model, BioClip, recognized that. I was telling Nick, it learned how to, it's got data that identified Darwin's finch based on their. These models can be really powerful. I'm trying not to get like too overboard, too exuberant, how power they have. Sure. I don't think we can ignore them. I mean, from large language models, all the different algorithms, if we can put it to good biological use, and to, let's say, so a lot of the examples that we have in minnows, we know that across the distribution of minnows, there's diversity as well. Cryptic species, hybrids, hybrid zones. Okay. That's a challenge. We have to train it, see if an algorithm can recognize the differences. Sexual differences, hybrids, cryptic species. It's all possible. That answers your question?
Yeah. All that I can say at this point is if you can get this thing to independently recognize like male and female serratioid anglerfishes, that will be incredible. Like just that I would be so stoked for that. And it would also really, really prove your point.
I can't imagine a more bizarre.
Whale fishes would be up there as well, but yeah, I mean something just completely insane.
But you know, it's a fish. It's a fish. You try to model recognize any fish, like flip that, then it'll know how to orient itself to it and do all the
Thank you.
other questions. I thought surely I'd give some doubting taxonomists in the audience. I don't know.
Yeah, and then there's numerical formula to differences. When we describe species, then we use skeleton, we use morphological features that aren't on the external.
You're right.
And
It's a very incomplete.
yeah, so it'll tell you there's differences, but it won't tell you the diagnosis.
Okay, so when this project, I have an answer. When this project was conceptualized, so it was first conceptualized with that beach eater. That was funded just only 2 years. We said we were going to do of the species that we were studying externally. And we were going to draw on those very graphic images outlines. Because you know, you can trace the outline, right? It's the part where you have bone that show up in the image, and then there's the outline around, right? And that's it's flounder is a flounder, whatever it is. That's good data. We wish we were doing this to be. We can't, we hate the fact that we can't do 3D with the fish of the fish. We've got the solution to that. This is a project Brian's gonna lead, is 3D surface scanning, but he also does 3D. He also does CT scanning. And so, you know, when you get stuff like that online, you're gonna get every detail about the morphology of the fish, internal image. So this is kind of like the rollout. Can we do this? how challenging. What are the problem areas, how, that kind of. One more thing came up at this HDR student. Paul Maybe was there, he dropped something. Paula may be presiding over Pinnacle Steam Beast or old libraries. And they built these anatomy ontologies, all kinds of fun, they want to let people, all the other kind of ontologies. For the fishes first, teleops first, but then all fishes. And what the ontologies do is tell you like the relationships, parts, different things like skeleton items, components. And you know, how we should think about it. Like this is a such and such, it's related to this trait, you know, they have codes and whatnot. At the end of the meeting we just had, we want to create a group of people to work with something called ontology AI. Ontology AI. So it's like, the idea is that if we add ontologies, like of any kind, gene ontologies, environmental ontologies, there's a bunch of them. We can train the models to work more quickly and do a better job. It's a lot more information. And it tells, this is like the explainable, right? It's like we're telling it what the relationship works. We're using biology to inform, that's knowledge guided machine learning, we're using that to inform, like, okay, right, this is the species, what it's supposed to relate to, we can establish this group with the biology. So this is gonna be an essence. Probably not involved, but it could involve any one of you, you know, who wants to get involved. And it's really cool. And you know, something that hopefully, I know that we'll get a lot more involvement if you can do the images and things you're working. So just wait. Not long. Fish air is pending. If it gets funded, or when it gets funded, or maybe before, I don't know. We're gonna put an imaging method, the best way to image specimens for this. And then you can start imaging specimens, actually, put them. The other things, CT data, radiograph, I had big file cabinet for radiographic images. Brian's gonna buy a scanner and just scan them. And so we have all those radiographic images, we have all of these things that radiograph. That's a huge resource radiograph that we can learn. We're gonna learn. I can recognize the fish in a radiograph, but I can't get all the details in a way that, you know, like a machine can. So. Anything else? Been a pleasure. I hope we get the STAR grant. This is the one I applied to prove the concept. And I hope that it convinces people this is a good idea with some get teams involved in or anyone else involved in, you know, see where it goes.
I love fishing if you get a grant.
So Brian's gonna take over my since you take over my grant. He's not gonna go fish. I'm going fish.
Well, let's,